Hi everyone. In the immunology subject, we were discussing about types of antigens and antibody reactions. In that also we started off discussing about labeled immunoassays. So the labeled immunoassays are of three types, that is enzyme linked immunosorbent assay, RIA and immunofluorescence. The RIA and immunofluorescence we discussed in the previous part, and in this part we are going to discuss about the ELISA, which means enzyme linked immunosorbent assay. So let's begin. So ELISA is a highly sensitive, highly specific, less expensive and safer technique used in serology to detect antigens and antibodies. So because of its highly sensitive nature, because of highly specific nature, and along with it less expensive and safer technique, we are going to use this ELISA widely in application. Then this technique or this test, an enzyme conjugated with an antibody reacts with a colorless substrate to generate a color reaction product. So in the previous part, we have discussed that if we are going to use a radioactive substance, then we name it as a radioimmunoassay. If we use the fluorochrome dyes, then we call it as immunofluorescence, isn't it? In the same manner, in this ELISA technique, we are going to take an enzyme which is conjugated with an antibody and reacts with the color substrate to generate a color reaction product. Such a substrate is called as chromogenic substrate. Okay. A number of enzymes have been employed for ELISA technique, including alkaline phosphatase, then horseradish peroxidase is another enzyme, and beta galactosidase. So these are the three main enzymes that were used or employed in the ELISA technique. And as I told you, the ELISA technique is used for the detection of many infections of viruses, bacteria, fungi, parasites, drugs, toxins, food allergens, etc. So coming to the principles that on which it is going to be of working is mainly two. One is antibodies and some antigens can adsorb, that means they can be imbibed or adsorbed to a solid support such as polystyrene or polyvinyl or polycarbonate wells or membranes or polyacrylamide paper. So this is going to be of like a tissue paper one, but they are going to be of made with all these substances. Then the second one, antigens and antibodies can be linked to enzymes. So that's why we call this as enzyme linked immunosorbent assay, with the resulting complex still fully functional, but both are going to be of immunologically and enzymatically. Okay. Now a number of variations of ELISA have been developed that allow qualitative detection or quantitative measurement of either antigen or antibodies. Okay. So this is all about the introduction and the principles, how it is going to work. So let's see the types of ELISA. So we are having the indirect ELISA, so then sandwich ELISA, and then comparative ELISA. So altogether we have the three types of ELISA. So let's begin with the indirect ELISA. Indirect. So we are not directly allowing the reaction to occur. So this is the indirect ELISA. So in this test, what is happening? So you can see here. So in this test, the antigen is going to be adsorbed onto the inner surface of microtiter well. Okay, so the wall of the microtiter well is going to be adsorbed with the antigens that we added. Now the test serum, that is patient serum, is added and incubated. So what happens during the incubation? If they are going to have any antibody related, obviously the antibody binds to the antigens. The presence of the antibody bound to the antigen is detected. So how can we know that whether the antibody is going to be bound to the antigen or not? So that is going to be detected by adding an enzyme conjugated antibody, anti-antibody. To this antibody, again we are adding another antibody which is conjugated with the enzyme, that is called as anti-antibody, which binds to the primary antibody. So now we are going to wash in between, we are going to wash with the buffers, okay, which will remove the free brown things. That means either the free bound antibodies or the free bound antigens or the free bound enzyme conjugated antibodies, whatever it may be. Now, and a substrate for the enzyme is added. So you have to watch, and finally what we are supposed to do, we are going to add the substrate also. Now the amount of the color reaction, so here you can see some sort of a color reaction product that forms is measured, or what we call it as a specialized spectrophotometric plate reader called as ELISA reader. So in that ELISA reader, we can measure the absorbance of all the wells of about 96 wells plate is a less than a few seconds. That means at a time we can have the samples to be detected of about 96. Indirect ELISA is used for the detection of antibodies to HIV and as well as for the rubella virus. So these are the things that we are going to discuss in detail. That means in the indirect ELISA. Then moving to the sandwich ELISA. Sandwich, so obviously in between the two breads. In the same manner, here the antigen can be detected or measured by a sandwich ELISA technique. So what is happening in this technique? First of all, the antibody is immobilized on a microtiter well. Okay, yeah. And then a sample containing antigen is added and allowed to react with immobilized antibody. Now again you are supposed to wash the well, and then an enzyme linked antibody was added, which is going to react with this antigen. So after an incubation, again it is supposed to wash to remove the excess of unbound antibody enzyme, that is enzyme linked antibody. Now the substrate is added. So once the substrate is added, if this complex is there, the substrate is going to be broke down, giving rise to the color reaction. And that color produced, that means the color product, is going to be measured in the ELISA reader. So this sandwich method, why we call it as a sandwich method, means the antigen is between the two antibodies looking like a sandwich. Maybe that's why this name was given as sandwich ELISA method. And this sandwich method is mainly used in the detection of HBs antigen, that is hepatitis antigen. Whereas the indirect is mainly used for detection of antibodies to HIV and rubella, whereas sandwich is mainly used to detect the antigens like HBs antigens. Then what is the third type of ELISA? That is competitive ELISA. What that is called as competitive. So here there will be a competition between the what we call it as two antibodies for the antigens to attach. So that's how the name maybe came as competitive ELISA. So here comes the picture where you can have the clear idea. So it is another version, that is competitive ELISA is another version of ELISA for measuring the amount of antigens. So what is happening in this technique? The antibody is first incubated with the antigen in a solution with a sample containing antigen. That means separately we are going to incubate them. Now the antigen antibody mixture is then added to an antigen coated. Already antigen was coated here. It is added to an antigen coated microtiter well. Now the more the antigen present in the sample, the less free antibody will be available to bind to the antigen coated well, isn't it? So what happens? The addition of an enzyme conjugated antibody specific for the isotype of the primary antibody can be used to determine the amount of this primary antibody bound to the well, as an we discussed in the indirect ELISA. Okay, now what is happening in the competitive ELISA? The higher the concentration of antigen in the original sample, the lower will be the absorbance. So if this is going to be more in number, this can't bind here. So what happens? It is not going to bind to it. So that's how we are, if this antibody is going to be more in number, then we can measure. So that is how the higher the concentration of the that is antigen in the original sample, the lower will be the absorbance. Why the lower? They can't bind to this well, so we cannot get the breakage of this enzyme substrate with the help of this one, which is not bound to this one. So it's an interlinker one. Okay, so here there is a competition between the unbound antibody and the bound antibody. So that's why this was named as competitive ELISA. So how many types of ELISAs that we have discussed? Three types. One is a competitive ELISA. Okay, then we have discussed about the sandwich ELISA. Then we have discussed about the indirect ELISA. Okay, thank you.